Hello. It's good to see everybody here today. What a pleasure to be with you in this beautiful city of Moscow at this health event. It's really nice to be with you. I'd like to thank the representatives who brought me here, Marina, who's over there, and thank you for arranging for me to be here. So what I thought I would do today is really give you an overview of how we actually grow tissues and organs and how this field is really advancing. And we do a lot of this work at this institute, where this whole building is dedicated to actually bringing these technologies all the way to the patient. This is a painting that hangs at the Conway Library at Harvard Medical School, and it shows the very first time an organ was transplanted. And this is back in 1954. And you can see the surgeon in the background getting the organ out of the donor. Well, in the front, you see the other surgeon, Joe Mary, actually getting ready to implant the very first organ into a patient. And this was back in 1954 in Boston. And the organ was a kidney. It was a kidney that was the very first organ ever implanted. And so many lives that have been saved by this one single event, but still so many lives that need to be saved because of the organ shortage and the organ rejection. The fact is that we are really at a major deficit of organs. In the last 10 years, there has been a doubling of the number of patients on the transplant list, while at the same time, the actual number of transplants has increased less than 1%. And that's because we're living longer. And the longer we live, the more our organs will fail. So wouldn't it be great if we could regenerate ourselves? Is that possible? Well, actually, it is possible in nature. This is a salamander, and this is a salamander limb. And this limb had been injured, and within 2 weeks, this whole limb was able to regrow. So the question is, if a salamander can do it, why can't we? Are we able to regenerate? The fact is we're always regenerating. I don't know if you know this, but the cells on your skin are turning over about every 2 weeks. The cells in your intestine are turning over also about every 2 weeks. The cells in your brain and your bones are turning about every 10 years, and so are the cells in your brain. So you're constantly regenerating. The challenge is that you do not regenerate when there is injury or disease or scar. That's when the body and the organs stop regenerating. And that's where this field comes in, this field that we call regenerative medicine, which is a field that brings together all these areas, cells and scaffolds, and enabling technologies to bring these technologies all the way to the forefront. For example, how about using cells? This is some of the studies we're doing right now, where we go to the patient, we take a very small sample of muscle tissue, we then process the cells, and we're then able to place those cells in the area where there is injured muscle. And just for example, 5 weeks ago, we started using these muscle cells for shoulder injuries at the time of surgery. So when the surgeon is actually going in to do a rotator cuff injury in the shoulder, we're able to implant these muscle cells to give that patient strength in that muscle. Now, these are the patient's own cells, the very specific muscle cells that we're expanding and putting right back into the body. And we've used it now for several different applications. We've also done it for skin. This is actually a clinical trial that we did at Wake Forest, where patients with burns, if you have a deep burn, that can cause scar. So what do? We can go to the operating room. We can take a very small sample of tissue from the patient's own skin. We can process the cells, and we just spray them over the burn area. And this is now phase 3 clinical trials in the U.S., moving into actual patient, and it's been treated in hundreds and hundreds of patients using these techniques. We can also use cells and scaffolds together. So what is a scaffold? Well, think about a strategy that we use to repair larger defects, where you have cells and scaffolds together. So this is actually a large defect. What you see here is an X-ray, and you're seeing this bladder and this tubular structure that's missing called the urethra, that is a channel that brings the fluid all the way outside the body. And we cannot repair this with just cells alone, because you need a scaffold and the cells. So what is a scaffold? A scaffold is very much what you see when you're putting together a building. The scaffold is the frame of the building, right? And you're putting the cement on the scaffold. Well, the cement that we use are the actual cells, and the scaffold is the material that we use. And these materials are very similar to the materials that we use for stitches in surgery. They're fully degradable. They totally go away on their own within a few months. But they're safe for humans, they're friendly to cells, and we use materials that replicate the biomechanical and structural properties of the tissue being replaced. For example, if we're trying to replace a piece of bone, that's going to require a very different material than if we're trying to replace a blood vessel, where the material needs to be nice and soft and elastic. So we use about 20 different materials that we mix and match to create the structure that we need for that particular patient. So for example, here's that same patient that I just showed you, and you can see this large defect, right? So we're going to use the scaffold and the cells. So how does this work? We go to the patient, we take a very small sample of tissue, less than half the size of a postage stamp. We expand the cells outside the body. We then create the scaffold in the shape of the organ, in the shape of a tube. We coat the outside with the muscle cells, the inside with the lining cells, put it in this oven-like device, and then put it right back into the patient. The whole process takes about 30 days. So here on the far left, you see that patient's X-ray. On the second panel, you see the actual material without the cells. On the far panel on the right, you see the material with the cells together. And here you see that patient with a fully regenerated urethra just 2 months after the biopsy was obtained. So you can see here that this fully regenerated urethra, and on the X-ray on the middle, this is 6 years after the surgery was performed, and this was published in this journal, and we are now moving forward with more clinical patients. Same strategy for blood vessels. This is what we call a bioreactor. We started to construct these bioreactors back in the 1990s so we could condition these blood vessels to do what they're supposed to do before you implant them back into the body. So what we do is the same strategy. We take this scaffold. It's a tubularized scaffold that we make with these degradable materials. The materials totally go away on their own within 6 months. We place the cells on the material, and then we exercise this material with the cells until it forms this vessel-like structure that we can then use to replace vessels in humans. This is what we call a carotid artery. That's the artery that goes from the neck to the brain, that was replaced experimentally using these techniques. These are heart valves. Now, we have not implanted these in patients yet, but just to show you how we do this, same as the blood vessels and the urethras, same strategy. We take these scaffolds, we construct them. The scaffold has the same properties as you would have on the organ itself, so the materials that we use replicate the structural properties of the tissue. We then are able to add the cells to it, use the bioreactors. We exercise these heart valves in these bioreactors. You can see the heart valve leaflets opening and closing, and we can then implant this back into the patient after this heart valve has been conditioned outside the body. Now, I'm showing you these in order of complexity. Flat structures such as skin that we talked about are the least complex because they're flat, they have basically one major cell type, and architecturally not as complex. We then talked about tubular structures like blood vessels or urethras or heart valves. These tubular structures are the second level of complexity because they're tubular, not flat. They have two major cell types instead of one, and they pump away at a design rate depending on the body's response. Hollow non-tubular organs are the third level of complexity because the organs have more architectural design than flat or tubular structures. There's more interaction with other organs, and the cells are more complex. So I'm just showing you this. Even this was, in fact, the very first organ that we implanted in a patient, but I'm showing you these in order of complexity. But same strategy. The strategy does not change. We bring the patient in to the hospital 4 to 6 weeks prior to their scheduled surgery. We take a very small sample of tissue from the patient, less than half the size of a postage stamp. We then take that piece of tissue, we tease the cells apart, 2 major cell types. We grow the 2 separate types separately and expand them outside the body. Once we have enough cells for our construct, we take a scaffold in the shape of the organ. We coat the inside with the inner lining cells. We coat the outside with the outer lining cells, put it in the incubator, and then put it right back into the patient. And this whole process, again, takes about 30 days. And we published that in that journal in our preclinical studies, and then we implanted these into patients, as you see here. Again, showing you that the scaffold that we build is no different than the scaffold that you would put together for a building, right? And instead of using cement and concrete, we're using cells to fill in the gap. And on the bottom right, you see the actual scaffold that we use for the bladder. And on the left, you see the bladder was implanted in the patient. And we published that also in this medical journal. By the time that we publish this series of patients, we have already had over a 5-year follow-up, just like I showed you with the urethras as well. This is another organ that we targeted. In this particular case, this patient was not born with this specific organ. There was a congenital abnormality. So now we're building an organ where there was none. So how do we do this? These patients are born with a syndrome where they do not develop this organ normally. But you can see in this X-ray the rectum on the right, the bladder on the left, and there's no vaginal organ in the center. We then take a small biopsy of the rudimentary structure. We expand the cells outside the body, create the construct, and plant it right back into the patient. And you see here on the far right, you see the rectum. On the far left, you see the bladder. And in the center, you see this new engineered organ that was created using these techniques. And this was also published. By the time we published this paper, we already had over an 8-year follow-up on these patients. And these patients, after the 8 years, many patients asked, well, why did you wait 8 years to actually publish this work? Because most patients that require typical replacement or repair, they use a piece of intestine or a piece of skin, and they tend to fail all the way up to year 5. They wanted to make sure we went all the way past 8 years to show that the technology really did work long term. And these patients continue to have normal function long term. So what about solid organs? By far, the most complex organs are the solid organs. And that has to do with the fact that there's so many more cells per centimeter than any other tissue type. The fact is that because there's so many cells, you need a lot more nutrition. And when you need a lot more nutrition, you need more blood vessels. And you need finer blood vessels. And when we started this work about 30 years ago, we really did not have the technology available for us to create solid structures. And that's when it occurred to us, why don't we just use discard organs? Let's say we take a discard organ from people who die, and they're not going to use those organs anymore, and we can reuse them. We can recycle the organs by using just the mold of the organs. So what you see here is a liver that we took, and we use very mild detergents to wash the cells away. And then 2 weeks later, you're left with something that looks like a liver. You can hold it like a liver. It feels like a liver, but it has no cells. But you are able to preserve the blood vessel tree. So what you're seeing here is just the scaffold, the skeleton of the liver with no cells. But the three-dimensional architecture has been preserved. So we can now perfuse those blood vessels with the patient's own blood vessel cells. We can then put in normal liver cells, and we end up with three-dimensional solid structures made out of the liver that function very similar to a liver in many of its features. Same thing for kidneys. In fact, kidneys are the #1 organ that tends to fail. If you look across all organs, 80% of the patients who are on a transplant list are waiting for a kidney. 80%. So here we used a little bit of a different strategy. What we did here is we engineered these miniature kidneys, but we used artificial materials. We didn't use a discard organ to do so. We created the three-dimensional structure, and we were able to show that we could create all the elements of these kidney units. And that was, we showed that we could do this with cloned cells. And that's why you see here successful transplantation of cloned cells. And so what we do now is we can go to a patient today, take a very small needle biopsy of their kidney. And this is done with a minimally invasive approach. So we go into the patient, we just do a very minimally invasive quick needle biopsy where we take a very small piece of tissue from that kidney. We can then grow those cells outside the body. Those are the patient's own normal kidney cells. And we can then insert them back into the deceased kidney. And this is showing you, on the far left, in a preclinical model, what happens with this sole nephron unit that's there. You don't have that many nephron units because the kidney is already deceased. So you only have one nephron. You know, you have these very spare kidney functioning units that are shown here with the circle in red. So what we did is we inserted all these new kidney cells, and then you had all these new kidney units that started to sprout up and grow within the kidney. And so by doing this, we're able to then reboost the function of the kidney, if you will. So let me tell you a little bit of information about kidney failure. Now you may think that when you have kidney failure, it just happens. But that's not really what happens. What really happens is that your body's organ starts to decrease its function over time. So let me give you an example. A patient comes in and they are playing tennis every week. Every week they go out and play tennis and they're doing perfectly okay. But all of a sudden, one day they go to play tennis and they get chest pain. So they rush the patient to the hospital, they take him to the emergency room, they do a cardiac catheterization, and then they find that the patient's kidney vessels are 90% obstructed. 90%. Guess what? That patient never had pain when the blood vessel was 60% obstructed, or even 70% obstructed, or even 80% obstructed. That patient was doing just fine, playing tennis every day. They only had chest pain when the blood vessels in their heart were over 90% obstructed. And the same thing happens to a patient who has lung disease. They show up to the doctor when they're short of breath, when their lung is over 90% diseased, and the patient with kidney failure also shows to the doctor when their kidney with swelling and difficulty with their bodies once their kidney is over 90% gone. Which means that nature has given you a 10x reserve. Nature has given you 10 times what you need in every single one of your organs, 10 times. It means that you have to blow 90% of that organ for you to get into kidney failure. So we try to do with a lot of these technologies, the key is to actually try to start early and treat patients early before they show up with the end-stage kidney failure and reboost them. So what do we do with the kidney? So this is one of the technologies we also have in patients. And what we're doing right now is we're in clinical trials for these patients where we have 10 centers throughout the U.S. In phase 2 FDA human clinical trials where they're getting a biopsy of their kidney, they're growing the cells out and getting the insertion. And what they're showing with these patients is that these patients that were on their way to dialysis, kidney failure, they basically did not get there. They basically were able to be rescued, and that's now in progress. So what about other technologies? So I've shown you now how we can grow flat structures such as skin, tubular structures such as blood vessels or heart valves, hollow non-tubular organs like the vaginal organ or the bladders, and we've been able to grow solid organs, some of which we have been able to implant the structures into patients. About 16 years ago, we decided that we really needed to scale this up. We needed to scale these technologies up in a major way because we were making these by hand, one at a time. And the goal was, how do you automate the process? How do you scale it up? And that's when we started with 3D printing. This is an inkjet cartridge, and instead of using ink, we started to use cells. And instead of using materials, we started to use these hydrogels that would go through these cartridges, and these would go one layer at a time. So printing two-dimensionally over and over and over again to create a three-dimensional structure that you see here, a miniature heart. And within a few hours, this heart would start to contract, as you see here. These are heart cells contracting. And even though we showed that this was possible, these structures did not have the structural integrity to be implanted inside the body. So what we ended up doing is we started to develop more sophisticated printers. These are printers that actually we built over a 14-year period. So we spent about 3 years doing the work with the desktop inkjet printers, and we then spent the next 14 years developing these printers specifically to create tissues and organs for patients. So this printing system has 5 features that make that possible. Feature number 1 is that these printers have extremely small nozzles. Very, very small nozzles. The nozzles are so small that they can go down to 2 microns. That's about 2% of the diameter of one human hair. So you just take one of your hairs and you look at it and you can picture something that is only 2% of that. Only 1/80th, about 1/80th to 1/90th the diameter of your human hair. That's how small these nozzles are. That's the first feature. The second feature is that it gives us precision. It allows us to lay the cells exactly where they're needed. And by doing so, we can recreate these cells one at a time and place them right where they're needed every time. The third feature is these bioinks. These are the inks that go through the nozzle. And what you want is you want this ink to go with the cells through the nozzle without damaging the cells. So you want this ink to be a liquid as it goes through the nozzle. Once it leaves the nozzle, you want it to be more like a gelatin so that when it hits the platform, it retains its structural integrity. And once it becomes a gelatin, then it starts to harden like a normal tissue. So that's the second feature. The third feature is these microchannels. These are microchannels that come in to give nutrition to the central portion of the cells, a highway system, if you will, that'll bring nutrition with blood-like substances all the way to the central core of the structure that we're printing. The fourth, and this allows us to create human-scale constructs and to print these as needed. And finally, the final feature is to use the currently available 3D imaging software that's already out there that allows you to configure what these organs look like inside the body, and it allows us to actually create a structure to fit specifically that organ. These programs are currently available in every major hospital where you can go in and you can have an X-ray done, and three-dimensionally they can configure what your organs look like. This is actually showing you here the specific organ. We can then go and we created our own software program where we basically have our own software program that then takes this digital data from the X-ray, and it can then use that data to drive the nozzle so the nozzle prints the structure specifically for that patient's defect. And we published that work back in 2016. So this is our newest printer, where we actually bring the printer to the patient's bedside. So the concept here is if a patient has a defect, you're actually going to be able to bring the printer to the patient's bedside. You're going to scan the area, and then you're going to go back with the printer and print what you need layer by layer on that same patient. So on the top left, you see a pictorial video. On the top right, you see the actual printer that we use for this technique. Here's another pictorial video. You go and scan the area. So the area gets scanned. That information gets transferred digitally to the software program that then programs the nozzles to print right where they're needed. What else have we done with the printer? Over the last 7 years, we've used the printer to create what's called the body on a chip. And what we did here is we combined the printing technologies that we developed with what was currently available through the computer industry with microchips, and what was available in the field of medicine with biosensing, to create miniature organs that we can print on a chip, or we can create on a chip, where all these organs can come together on these chips, and they can start to function one with the other in a miniature way. So basically designed in the body in a chip. And you know, this is still in development in terms of the printer, in terms of laying these correctly with good viability, but we can place these miniature hearts, miniature lungs, livers, blood vessels, etc., into these microchips the size of a slide, the size of a very small coin. Why is that important? The reason why that's important is because currently the way that drugs are developed are by testing these drugs in animal models and cell lines. That's how these drugs get tested. So the way that the pharmaceutical industry works is they develop a target and they develop a compound for that target. Let's say to treat high blood pressure, right? You're going to treat high blood pressure, so they're going to develop this pill that you're going to take every day to control your blood pressure. But they're going to test this pill in cell cultures. They're going to do cell cultures using cell lines, or they're going to use animal models. The challenge is that animal models don't necessarily replicate what humans do. That's the first challenge. The second challenge is that cell cultures are just cells. They're not really organs. So what do we do about that? We can create these miniature organs in the same manner. We create these miniature hearts. They're about the size of a pinhead each, and we can put them on these chips, and they react just like your normal heart would react. So for example, if we give these hearts drugs that will speed it up, they'll speed up the heart. If we give drugs that will slow down your heart, and we give it to these miniature hearts, it'll slow down those miniature hearts. So these are doing the same thing that your body does. We're replicating your organ with this miniature structure on a chip. So why is that important? We can test these. So, for example, here's a drug that was in the market all over the world for 11 years called Hismanol. This was an antipsychotic medication, also used as an antihistamine for colds and allergies. And this drug had been on the market for 11 years before they realized that there were enough patients out there that were actually having a heart block, and they connected all these patients to this one drug. It took 11 years for physicians and the healthcare system and patients to realize that it was this drug causing the problem. So this drug had been tested in cell lines, no toxicity was noted. It had been tested in animal models, no toxicity had been noted. It had been tested in phase 1, 2, and 3 human clinical trials, no toxicity noted. And it took 11 years. So guess what? We took this drug, put in our chip, and within 2 weeks, we knew that this drug was toxic to the heart with the same mechanism of action. We can place these in what we call these microchips, and we print these chips. You can see here on the bottom left this microchip that we printed, and this microchip has ovaries, kidneys, and liver within this one chip. And you can see the blood-like substance going through the chip, so we can actually measure just like in the regular body, right? Your blood is going through your organs. We can mix these organs up, and we can actually start testing them. Here's another example. This is Resolin, an anti-diabetic drug. This drug had been in the market for 3 years. Much more tragic story. This drug had been tested in animal models, no toxicity noted. Phase 1, 2, and 3 human clinical trials, no toxicity noted. The drug got released into the marketplace, and within 3 years, there were 63 deaths due to liver failure. So we took the drug, tested in our system, and within 2 weeks, we knew the drug was toxic to the liver, with the same dose response, with the same mechanism, and with the same pathology that we would see. Now, why is that? Why is it that it took 11 years for that other drug before we could figure out that the drug was causing problems? It took 3 years for us to realize this drug was causing problems. And we can take this drug and test it in our body in a chip, and within 2 weeks we know what's going on. Why is that? That is because all of us process drugs differently. We're all genetically different. So when you take a drug, you may be taking the same drug, but all of you are processing that drug in a different way. You all have different diets. You all have different things that you eat. You're under different conditions. And it takes, and you have all this confusion, all this noise that happens where the drugs, you're not really seeing what the drug really does to the organ. And with this system, you're taking all that noise away. You're basically testing the drug against the organ and giving you that toxicity right away. So the development of this system, what we call the body in a chip, is really to help develop drugs for the marketplace, to help reduce toxicity, but we're now using this system for patients with cancer. How are we doing that? If a patient develops cancer, we can now take a very small piece of tissue from the cancer and build a cancer on a chip in the body of the patient, kind of a body in a chip with the cancer. And we can now test those chemotherapy agents on that chip before the patient gets that drug. So let me give you one example. There was a patient, and we now have this in 5 different clinical trials at our institution with our oncology team, Kostas Ganopoulos and others in our group, Shai Soccer. There's a whole team. We have 5 clinical trials going on: brain cancer, breast cancer, colon cancer, pancreatic cancer, myeloma. And what we do is we take a very small sample of tissue at the time that the patient gets the tumor, we put on the chip, and we test it. There was one patient who had a melanoma. This patient had 6 months of chemotherapy, and the tumor kept growing. Several $100,000 later, 6 months later, it kept growing. The patient, they got switched to another chemotherapy regimen. 6 months of treatment, the tumor kept growing yet again. So now $500,000, and a year later, no success. So they called us up and they said, we want you to try this. So our team basically tried this, and they found one drug that had not been considered. So they got permission to use the drug. The patient received the treatment. Within 2 weeks, the patient called the doctor back saying, for the very first time, the tumor is shrinking. So the key is to try to figure out how to treat you before you get the treatment. Everything we've talked about thus far, we've used the patient's own cells, right? When we made those, the heart valve or the kidney, we always go to the patient. We take a small sample of tissue from that organ, and we grow and expand those cells, and we put them right back into the patient where those cells know what they are and they're not going to reject. But what if you don't have that organ? Let's say that you were born with only one kidney, you get into a car accident, and you lose the only kidney you had, and now you don't have any kidney anymore. What are you going to do? Well, that's where stem cells can come in. A stem cell is basically any cell that can be converted to other things. So right now, what I've shown you is that to make a kidney, we take a kidney cell from the patient to make that kidney. If we're going to make a urethra, we take a urethral cell from the patient to make that organ. If we're going to make a blood vessel, the same thing. But a stem cell, we can take one cell, and that one cell can become a kidney cell, or a blood vessel cell, or a lung cell. And there are two major types of stem cells out there. They're what we call human embryonic stem cells and induced pluripotent stem cells, and these are very powerful cells. Very powerful. They grow rapidly. They can turn into anything. But I don't know if you know this. How many people here know that these cells can form tumors? Raise your hands if you knew that. Very few people know that, actually. Only a couple of hands. Let me just tell you. You knew that, yeah. If you do, raise your hands if you knew that these cells could form tumors. Yeah. So not many hands go up. But basically, the bottom line is that both human embryonic stem cells and induced pluripotent stem cells, these cell types, they form tumors. Very little known fact, but it's true. That's why they have such a difficulty getting into patients. So that's one of the challenges. On the other hand, you have adult stem cells that you can get from cord, you can get from fat, you can get from bone marrow. And these cells will not form tumors, but unfortunately, you cannot get these cells to grow outside the body in large quantities. They don't expand very well. So about 17 years ago, we started looking at an alternate source of stem cells, and we basically just postulated that perhaps there was a stem cell type that was present in the amniotic fluid and the placenta. So the baby in the developing womb is surrounded by the fluid. That's the amniotic fluid, which is what when the water breaks. And then the newborn in the womb is the baby in the womb is also surrounded by the placenta, which is also called the afterbirth. And we postulated that perhaps these tissue sources had a stem cell within them. And sure enough, we were able to find that there was a very, very powerful stem cell that was present in the amniotic fluid and the placenta that could go into all three major tissue types, just like human embryonic and induced pluripotent stem cells. So it's a new stem cell class. We can get it from the placenta or the amniotic fluid or both. You can get it before or after pregnancy. These cells, unlike human embryonic stem cells and induced pluripotent stem cells, they do not form the tumors. And like adult stem cells, you can get them to grow, and they go into all three germ layers, which are the major categories of tissues that develop other tissue types. And these cells could be expanded to large quantities and either stored for self-use or stored as a bank for other patients if you match them. And we published that work in this journal, and basically is a new type of a cell that is now entering many different clinical trials. So these cells are now being used extensively for various indications around the world. So to summarize then, what I've tried to do for you today is really give you an overview of where these technologies are. We are basically working on about over 40 different tissues and organs. We have about 15 of these that we have already implanted into patients. And this year, we're going to implant tissues or therapies number 16 and 17 coming out of the Institute, all in different areas. Our goal is really to bring more of these technologies to patients and to keep expanding the number of patients that receive these technologies. We also have this major program that's funded by the Department of Defense and the Army and the Navy and the Marines for our wounded warriors. This is a $75 million program. We're on phase 2 of the program. This is now year 10. So they've invested 85 plus 75 million plus several other tens of millions. So they've invested about $200 million to date over the last 11 years for us to bring these technologies from the bench to the bedside. And under this program, we basically now have 18 clinical trials that have been deployed through the armed services with the purpose of reaching our wounded warriors in this area of regenerative medicine. So to summarize then, what I've tried to do for you today is really give you a broad overview of where this field is at. We've talked about using cells alone, like for skin, for example, with a spray, or injecting skeletal muscle cells for shoulder injuries. We've also used them for incontinence and other areas. We've talked about using cells and scaffolds together when you need to replace a structural defect, like a blood vessel or a urethra or other organs. We've talked about using several strategies where we recycle organs and we get rid of the cells to implant your very own cells on that organ. We've also talked about 3D printing as yet another strategy that we're using to bring these tissues to patients. We also talked about the levels of complexity. Level 1 being the flat structures such as skin. Level 2 being tubular structures such as blood vessels. Level 3 being hollow non-tubular organs like the bladder. Level 4 being solid organs. We've now been able to impact all of these 4 areas. So at this time, I'd like to share with you a very brief video of a patient who was treated with one of these organs. And this is from an interview he gave about 10 years after receiving his engineered organ.
I was really sick. I could barely get out of bed. I was missing school. It was just pretty much miserable. I couldn't, go out and play, basketball at recess without feeling like I was going to pass out when I got back inside. I felt so sick. I was facing basically a lifetime of dialysis, and I don't even like to think about what my life would be like if I was on that. So after the surgery, life got a lot better for me. I was able to do more things. I was able to wrestle in high school. I became the captain of the team, and that was great. I was able to be, the normal kid with my friends, and because they use my own cells to, build this bladder, it's going to be with me. I got it for life, so I'm all set.
That's Lucas Masella. As I mentioned, that was an interview he gave 10 years after receiving his engineered organ. He's now actually an executive in New York. I just had the pleasure of speaking to him about 3 months ago by phone. He's doing very well. He actually had sent somebody else that he wanted that had reached out to him. The fact is that these technologies still have many challenges ahead. I mean, I don't want you walking away from here thinking, boy, every problem has been solved. Also, I don't want you walking away from here thinking this stuff is so easy. You just put cells in the printer, you push a button, out comes the organ. Nothing could be further from the truth. Actually, these technologies take decades to develop. They're extremely complicated. You have to know the protocol time and time again before they can work. Once you have the recipe, of course, you can make it work and do right by them. The technologies are costly. We still have to make sure we can scale them up. But one thing is certain, and that is that these technologies do have the potential to make patients' lives better. And for us, that's the promise of regenerative medicine, to make patients' lives better. Thank you for your attention today. Appreciate it. I purposely left some time for questions, so we'll go ahead and open up to questions.
Good afternoon. Thank you very much for your amazing presentation. It is very clear, and thank you very much for your work. I have a very practical question. We have been waiting for your technologies for 18 years because 18 years ago, my granddaughter received a diagnosis of arthrosis of a face bone. Now she's 18 years old. She lives in London. She has gone to the United States for consultation. I'm not going to go into details since I'm not going to be able to explain the medical details. Now she grew up, and it is proposed for her to make a titanium implantation. The question is, should we wait a little bit more to try to grow this organ? As far as I understand, this is still very complicated. Thank you.
So basically, there are already bone substitutes that are being used experimentally. So there are actually clinical trials right now for bone substitutes. So I would definitely check into that. and see if that's available. But you know, because that's a bone that's you know, it's a little bit, you know, the shape is a little bit complex and it can be reproduced. So I would actually check into that and make sure that you can use some of the existing technologies. But bone is actually one of the things that has really moved towards the clinic faster, because you can use many different type of cells and strategies for bone. So I would definitely look into that before, you know, so of course titanium is the current standard, but there are things in clinical trials now that you can actually look for.
Thank you very much for this great presentation. I always listen to your lectures with pleasure. Kovalyov Vyacheslav, Deputy of the Lab Orthopedy and Regenerative Medicine of Russia. As I know, at Sechenov Institute, you also cooperate with this organization. What do you do there? You cooperate with Sechenov Institute. What do you do there? You cooperate with Moscow Sechenov University. Do you cooperate? Do you work there also? My question is, what do you do there? What is your function at Sechenov Moscow, Sechenov University?
It's a great university. We are working closely with them, and but we work with many different places all over the world. We have about, you know, we do an inventory of collaborations every December. So we just had the inventory actually, and we do active collaborations that our institute has. As of this December, we had over 400 active collaborations worldwide. About 300 of these are in the United States, and over 100 of these are outside the country. And the question is a very good question, and the answer is very simple, that we really do believe that these technologies need to be advanced all over the world. And there's so many good people and so many good centers doing this work, as you know. And the thing is that it's very important for us to share and to make sure that people can advance these technologies all over the place. And of course, being here in Moscow, it's a wonderful place to be and a great place to collaborate.
Hello, can you speak about how cells go to detergent organ? Because this is a complicated structure inside of the organ. How can the cell be transferred to this organ? How can they go to this particular place? Because cells, how can they penetrate inside?
That's a great question. So when we use these discard organs, what we're doing is that we're using these very mild detergents to wash the cells away. So you're left just with the skeleton of the organ. And then the goal is to actually, you perfuse, but you're retaining the vascular tree, the blood vessel tree. So we're perfusing the blood vessel tree with the cells, and then we're infiltrating the parenchyma of the structure with the other cells. And the cells really know what to do. That's one of the amazing things about nature, is that the cells really do have the ability to regenerate and do what they're supposed to do. Every single cell in your body has the potential to create a whole new you, right? With cloning. You know how cloning, you know, when Dolly was cloned. Do you remember when Dolly the sheep was cloned? So that was done with one cell. So basically, any one cell in your body has all the genetic information to create a whole new you. So how is that possible? Well, how is it possible that every cell in your body has that genetic information? It does. Every cell in your body is like I make the analogy, is like a piano with all the keys. And basically, what happens is that you play different songs. So if you're a heart, you're going to play a different song. If you're a liver, you play a different song. If you're a liver, you play a different song. But you're using the same keyboard. That's your genetic makeup, and that's why you can replicate. So the cells know what to do once you put them in the right environment. That's the bottom line. That's how the technology, the cells actually, reform and regroup just like they're supposed to create the structure.
Thank you very much for this lecture. I have a question about stem cells from this amniotic fluid and placenta. You have mentioned about this. Do I get this right, that these stem cells can be used for mother of the child, or do you use them for other people also? And if you use them for this mother of the child from whom you take stem cells, and what about men? How can we give the stem cells to men if they are not mothers?
That's a good question. So the way that we get, so basically the stem cells that we obtain are for the baby. So they're specific to the baby from the amniotic fluid and the placenta. And so what happens is you can just wait until after the baby's born and just wait until, the baby's delivered, then comes the afterbirth, which is the placenta. So you can just basically save a small sample of the placenta and save the stem cell population, and that now becomes a repository for that baby for the rest of their lives. Very much like cord blood, cord blood's being used where they offer you that service, but cord blood is basically right now being used pretty much for blood cancers, for leukemia and other cancers, and cord blood cells will not expand. So when you get one cord out of the baby, that cord will only last for cancer treatment till the child is a teenager. By the time they become a teenager, they already need transplant from 2 cords. So maybe I'm being too technical, so let me take 2 steps back. If you get blood cancer, there are 2 ways to treat it often for some of these treatments where you use stem cells. You can either get the cells from the bone marrow, or you can get cells from the cord that gets preserved at the time of birth. So those cords can be used either for that same child, if they get the cancer, or if they get to be over the age of 17, then they have to use a common cord from somebody else. Same thing here with a placenta. You can use those cells for that baby for the rest of their lives, with one exception. They expand, they keep expanding, so you can keep using the cells over and over again. And they do turn into other things. They go into all different tissue types. They can become bone or heart or liver. So they do turn into other things. They're not just for cancer. So you can save those cells for that particular baby, or you can actually create a bank where you can donate the cells for others, just like you do with cord. Yes.
Hello? What organ is more complicated to restore? Which organ? Yes, which organ is more complicated to restore?
Far, the most complex organs are the solid organs, like the heart, the kidney, the liver. They're by far the most complicated because the solid organs have many more cells per centimeter than any other tissue type, and it's much more complex. So the flat structures, the tubular structures, and hollow organs, so things like skin, blood vessels, or bladders, they are complex to make, but they do not have such a high requirement of nutrition as your solid tissue does. So that's still a major challenge, and that is why for solid organs right now the strategy we're pursuing is to make sure that we can get you away from the final failure from the organ at this point.
Thank you very much for this lecture. You said that it's very difficult to make this heart and liver. What about eyes, for example?
There's a lot of work with eyes. So there's work with retina, for example, that there have been stem cell trials for the retina that have been implanted for the retina. That we are working on cornea. for the eye. The optic nerve is very, very complicated. So that's that's analogous to the spinal cord. A lot of research, but nothing yet that can go to a patient. For the retina, there have been clinical trials done for injecting cells into the retina. Cornea has been engineered and is going to be going into patients. So it's different components of the eye that are being worked on at this point, not the entire eye. It's the different parts of the eye that are being worked on.
Спасибо.
Thank you very much. Thank you very much. This is a great lecture. I want to ask you about this. How do you avoid immune response to structural organs? You clean them, you make a construct out of them. Do you expect this immune response, and how do you cope with this immune response, if it is?
Yeah, great question. So the immune response. So basically, everything I've shown you today uses the patient's own cells. So there is no need for rejection, so for anti-rejection medications. And that's one of the things that we have really stressed from day one in terms of the work that we do. So if we are creating a tissue from your very own body, what we do is we take a very small sample of tissue from your very own body, the patient that's going to be treated. That's where we take the sample from. And those are the same cells that get expanded, and those are the same cells that we use for the construct. So when they get placed back into you, you don't have an immune response. You don't have a rejection to that tissue. So that's how we get around that problem, which is one of the advantages, really, of course, of regenerative medicine, that you can use a patient's own cells.
I know that you're very great in translation, okay?
Yes.
And science. Do you have any bioprinter in situ or not in situ which is certified for clinical use?
Yes, we do actually have one of the printers that we have. We have another technology that's based on laser capture, and and we have two printer systems that are actually certified to create tissues for human use at this point. So using two different technologies.
FDA approved?
Yeah, so that basically this is going through the process so that when these tissues get approved. So, so at the FDA, the FDA is not regulating the actual manufacturing devices. They regulate the manufacturing process. So the way that this works with the FDA is that what they're looking at is they're looking at the final product to make sure the product is safe. They're not going into the manufacturing facility and saying, well, you need to certify this incubator or you need to certify, but they do need to certify, you do have it within a certified facility that has those properties that allow you to create a tissue that's going to be sterile. What the FDA regulates is what you're implanting into the patient. So basically, that's what they're regulating. Yeah. By the way, that's Vladimir. So, this is a colleague of mine. We go back many years, who's done a lot of work in the printer, and he was in South Carolina, just one state below me. It's great to see you. Great to see you, my friend and colleague. Another expert in printing right here in Moscow.
Hello. Can you tell me, do you have some works on growing neurons of brain to treat Parkinson's disease, some neuron generative diseases? Maybe it's simpler to grow than organs.
Yeah, so basically, we are working on, that's one of the areas we said we would never work on was brain. By the way, it's Vladimir Mironov. That's Vladimir Mironov. One of the things we said we would never work on is the brain because it is so complex. But with time, you never say never, because that's what we said several decades ago. We are working on brain. We're making miniature brains with all the 6 different cell types of a human. We are working on them now. We just started that work about 7 years ago. In terms of, and that's for brain engineering, you know, where you actually create this structure. In terms of brain therapy for Parkinson's, you know, that's been done. That was done, you know, 25 years ago, where they were taking animal cells and put them into patients, and in fact, they had good outcome. The patients did have a good outcome. These clinical trials were done in Boston by a colleague of mine, Sam Elias, and others. And they did show that the technology really did work. The challenge was that there were not enough animals to have enough cells to treat patients, and it was a very labor-intensive process. I think there's going to be a lot of improvement in Parkinson's. The challenge with Parkinson's is that it's not a disease. It's not a disease necessarily of the tissue itself. It's a disease of the connectivity and secretion of all these tissues. So it's going to be a lot more complicated than you would think to actually replace a piece of the brain for Parkinson's. That's an area where cell therapy will be more amenable than to actually engineer a brain for something like Parkinson's.
Вот вы говорили
You said that you take a small piece of kidney and then you regenerate this, but then old kidney does not function practically. But then in regenerated kidney, you have more functioning parts. How can you get it? It was a bad kidney. How can you make a good kidney out of it?
What we do when we take these cells, we are really taking the normal cells from the kidney. So what happens is, in your any organ that fails in your body, any organ that fails, whether it's kidney, heart, liver, pancreas, what happens is that there's a lot of scar that gets formed, and when the scar forms, you do not have the regeneration of the cells or the tissue there, but you still have normal cells within the tissue. So you can still take that. So what we do is we actually select for the normal cells, and that's what we're using to expand. So even in a extensively diseased organ, like someone a 70-year-old patient with end-stage kidney disease, we can go in with a needle biopsy and still get normal cells from that patient. And those are the normal cells that we're using to treat the patient. And that's why those patients are, you know, stabilizing in terms of their kidney function. Now, one very important point, and I'm glad you brought this question up, because the important point is we cannot do that for genetic disease, right? So let's say somebody has cystic fibrosis of the lung. That's a genetic disease. You're born with it. So every single cell in your body, your cells have that defect within the cells. Or let's say muscular dystrophy is one that is a little bit more common. People understand muscular dystrophy because they stop walking and they have to be wheelchair bound. If I were to replace the skeletal muscle in that patient's own skeletal muscle is still going to have that defect. So what we're doing for all of these areas which are genetic defects like hemophilia, muscular dystrophy, cystic fibrosis, the strategy that we're taking is gene editing. So what we do is we take the cells from the patient, we then do gene editing, so we cut out the defective area, and then put in a normal area in, and we can then package the cells and put them right back in to the patient with the normal cells. So I didn't talk about that today because we're doing a lot of the research right now in that area, but that's the strategy. For patients with genetic disease, which are fortunately, genetic diseases make up less than 1% of all organ failure, which is a good thing. But the bad thing is these patients still need to be treated, and the strategy to do that is with gene editing again. Just to, again, let people know what that is. Gene editing means that that's the defect that patient has in every single cell, right? That genetic information, that cell that can create a whole new you. So what we do is we take the cell, and we basically, you know, with a little pair of molecular scissors, we cut out that defective gene out, and we then put the normal gene in. We use glue to patch it up, grow those cells, and then put them back in. So that's the strategy that we're working on right now. We have a few papers published in that area. There was somebody here.
Да.
Сколько по времени?
What time, how much time do you, so how long do you grow this organ? For example, very simple flat, so for 1 month. So how long do you do this?
Pretty much the strategy is interesting because for most of these, within a month, you can grow it. You can grow the structure within a month, whether it be skin or whether it be any other tissue type, because you just grow more cells to create the structure. But basically, the process is you grow the cells out, and once you have enough cells, then you start creating the construct. And then you let the construct sit in the bioreactor for about a week before it gets implanted. So for all these technologies, it's about a month to create. And thankfully, you know, pretty much for all these diseases, you can wait time before you actually reconstruct the patient, right? So even the patient who comes in with trauma, you need to stabilize them and make sure they're stable and infection-free before you repair them. So you do have time to wait for these patients for formal treatment. Sounds good. I think it's 6 o'clock, so the time is up. It's really great being with all of you today. Thank you so much for your attention. Thank you for having me.